So usually all your carbohydrates are going to have this structural formula, or a multiple of that. And so linkages between carbohydrates are called glycosidic linkages, and I think it might be better to memorize like all the different names for the bonds between monomers for different macromolecules. And so carbohydrate linkages are called glycosidic linkages. And so yeah, the main storage carbohydrate you should know for that are polysaccharides are starch, which is for energy storage in plants, and glycogen, which is in animals. And you have structural polysaccharides, which are cellulose in plants for their cell wall, and you have chitin in fungi, and that's also for the cell wall. And so yeah, your sugars are mainly your primary energy source in the body, and it also helps with structure in plants, the wall, and fungi's cell wall too. And so a crucial thing is that basically these are all carbohydrates, and they're actually all the same carbohydrate. So these are all like monomers, and which is glucose. So like the crucial thing is if AP biology, if they show you a diagram of a carbohydrate, you have to be able to recognize it. And these are all the ways they can basically represent carbohydrates. And so the next macromolecule are proteins, which are molecules with one or more polypeptides. And so a polypeptide is basically the polymers of amino acids, and the amino acids are basically the basic monomers of proteins. And so your amino acids are made of a central carbon with amino group, a carboxyl group, and an R group. And what's important about the R group is that that's basically the side chain, and it's super important because every single amino acid has a different side chain, and that's why there is specificity between different amino acids. Like that's why amino acids are different, because of the fact they have this side chain. And so amino acids can be hydrophobic, hydrophilic, acidic, or basic. And the important thing is you need to know what it's called between a bond between amino acids, and that's called a peptide. And so basically you need to know about the polypeptide backbone, which is linkages between amino acids, and they don't include the R group. And that's super important because when you're talking about protein folding, you definitely need to know what the polypeptide backbone is. And so you need to also know about globular proteins, which are spherical, and you have fibrous proteins, which are like long fibers. And the next thing you know is how proteins fold. So you have a primary structure, which are basically the amino acids linked together in a specific sequence. And you have the secondary structure, which are the hydrogen bonds between the polypeptide backbone. That's really important because secondary and tertiary structure differs in where there are hydrogen bonds and where there are different interactions. So the hydrogen bonds are between the polypeptide backbone, which means it's between the carboxyl and amino groups shown, which is basically the polypeptide backbone. And they form alpha helices and beta pleated sheets. You definitely need to know the different types of secondary structures, and that's alpha helices and beta pleated sheets. And then the tertiary structure are your interactions between your R groups, and that's basically your side chain, so it's not the polypeptide backbone. And so there are different interactions in tertiary structure, like hydrophobic interactions, sulfide bridges, which are between sulfohydryl groups, and then you have ionic bonds, and then van der Waals. So these are all, you don't really need to know specific, like what van der Waals is, but you should know like some examples of tertiary structure interactions. And also you have quaternary structure, which are the interactions between different polypeptides. And so if you have multiple polypeptides, then it's crucial to have a quaternary structure. Because not all proteins actually have a quaternary structure. It really depends on how many. If they have more than one polypeptide, then that would be called have a quaternary structure. But every single protein, no matter what, has a primary, secondary, and tertiary structure. And so this is kind of how like proteins fold. This is your primary protein structure, and then you have your secondary structure, your tertiary structure, which is due to the R groups, side chain interactions, And you have your quaternary protein structure, which is due to different polypeptides. And yeah, these are good examples of globular proteins and a fibrous protein. So collagen is long, and then myoglobin is more circular shaped. This is like your basic amino acid structure, which is your amino group, carboxyl group, and your side chain and a hydrogen. And so the important thing about proteins is that the unique shape of a protein determines its function. So you're doing usually a test why proteins are so in this case that's what you're going to use for proteins, because it's based on the shape and that's how it has function. And so chemical conditions of a cell change. If any chemical changes occur in the cell, such as pH or salt concentration, any of that, it can lead to denaturation of the protein, which means that proteins will lose their native shape, leading to the inactivation of the protein. So the protein basically can't function anymore because of the fact that it's denatured, which is due to maybe a pH difference or temperature difference, whatever. But if cell conditions are restored, so if they get back to normal, the proteins can renature and get back to their native shape, which is always due to folding. So denaturation and is due to folding, and then denaturation is just due to the conditions.
I have a question. So after it's denatured and it has to renature, does the protein have to go back to the Golgi apparatus, or is it gonna?
No, it doesn't have to. It can just be like anywhere in the cytoplasm. It could start folding there.
All right, cool.
Yeah, and then and then the next is nucleic acids, which are polymers made of monomers called nucleotides. So your nucleotides are your monomers. So there's basically a sugar, which is deoxyribose or ribose, depending on whichever nucleic acid you're talking about, and you have a phosphate and a nitrogenous base. And so the important thing is like the distinction between 5' and 3'. So 5' is basically the carbon of the sugar that attaches to the phosphate. And then 3' is basically the hydroxyl group attached to the third carbon of your sugar. So that's really important because if you're talking about replication, 5' to 3' is really crucial. Transcription or replication. So you have two types of nitrogenases. You have pyrimidines and purines. And so the difference is purines have 2 rings in the nitrogenous base. So that's basically adenine, and pyrimidines have 1 ring, which is basically uracil, thymine, or cytosine. And so, yeah, the 2 types of nucleic acids are DNA and RNA. DNA provides your directions for their own replication, and it also directs RNA synthesis. And RNA basically directs protein synthesis. And so, yeah, there are a lot of differences between DNA and RNA, such as DNA having thymine, it's usually double-stranded, and it has deoxyribose. RNA, on the other hand, uses uracil instead of thymine. It's usually single-stranded, and it has ribose. It has ribose instead of deoxyribose. And so the difference between deoxyribose and ribose is that DNA, the sugar is deoxyribose. The key word is oxy, which means it doesn't have an oxygen. So it doesn't have an oxygen in the 2' carbon, which is this carbon. If you can see my cursor. And then so the crucial thing also about DNA is that it's anti-parallel, which means if one strand is 5' to 3', the other strand, the same direction, will go 3' to 5'. And that's crucial for DNA. And so sugar to phosphate bonds are called a phosphodiester bond. And while you have, and then yeah, so basically, and then also there's a crucial phenomenon called Chargaff's rules, which shows that guanine is always equal to the amount of cytosine in a cell, and adenine should always equal the amount of thymine. And that also, if you have different organisms, they have different percentages in the bases of your nitrogenous bases, or adenosine, thymine, cytosine, and guanine. And so, because the strands are complementary, adenine always pairs with thymine, and cytosine always pairs with guanine. So you can always predict the sequence of one strand by looking at another, which is crucial for DNA replication because you can use one template strand to form another strand. And the thing is, nitrogen bases, they pair by hydrogen bonding. And this is really important. You need to know that A and T, they form 2 hydrogen bonds, and cytosine and guanine form 3 hydrogen bonds. And it's really important because if you're talking about, if you're basically, there's always this question that they talk about, like organisms that live in higher temperatures and how their DNA becomes super stable. Like, why is their DNA so stable compared to other organisms? And the answer is because they have more cytosine and guanine, meaning that they have 3 hydrogen bonds instead of 2. So I think that's a common test question, like asked about the stability of DNA. And also you have lipids, and the crucial thing about lipids is it's actually not a macromolecule. And the crucial thing between like molecules that are grouped into lipids is that they're all hydrophobic. And so it includes fats, phospholipids, and steroids. And so fats are made of glycerol and fatty acids, which is a hydrocarbon chain. It's also why your fats are usually hydrophobic, because you have a hydrocarbon chain that's made of carbons and hydrogen. And there are 2 kinds of fats, unsaturated fat and saturated fats. The thing is, saturated fats have a double bond. And so it's tight packing between fats, and then you also have saturated fats, which don't double bond. And that means that they are saturated with hydrogens. And that's a good way to remember like what's the difference between a saturated fat and unsaturated fat. That are saturated with hydrogens because it doesn't have a double bond. And so saturated fat could be like butter, which is able to pack closely together because the fact that there isn't a double bond. Then the main way that we can store fat is using triglycerides, which are 3 fatty acids linked to a glycerol molecule. And the thing about fat is fat can store 2 times as much energy as carbohydrates, which I think that's also sometimes what they test on. They sometimes ask you how much energy storage is in different macromolecules or like fat compared to carbohydrate. And the thing is, fats can help with insulation, and they're also energy, and they can help cushion vital organs. So fats aren't just used in storage, energy storage. And then so you also have different types of lipids like phospholipids, which are crucial in cell membrane structure. The thing about phospholipids is amphiphatic, which means they have a both nonpolar and polar side. And it's basically a glycerol attached to phosphate group, and that's basically why phospholipids are polar, because there's a phosphate group that helps make it polar. And you have your 2 fatty acids, which make phospholipids also partially nonpolar, so that's why it's both polar and nonpolar. And so you also have steroids, and steroids are lipids with fused carbon rings. And so the important thing about steroids is that a lot of them are hormones like estrogen and testosterone, and that steroids like cholesterol can also help with cell membrane flexibility. And I think I have slides on cholesterol later, so we don't need to go too in depth right now, but basically this is how a steroid looks like with your 4 carbon rings. And that's basically unit 1. I didn't any slides on water because I think it's pretty basic. So I think most people know everything about water. So yeah, we can jump into unit 2 if you guys have any questions.
Yeah, so can I ask?
Yeah.
Okay, so where does protein folding occur, like in the cell? What fraction? The nucleus, the cytoplasm, or the Golgi apparatus?
Yeah, so protein folding can actually happen either in the cytoplasm or the endoplasmic reticulum. So it kind of just depends on the protein. Like if your protein is being exported like from the endoplasmic reticulum to Golgi to outside the cell, then your protein's going to be folded in your endoplasmic reticulum. But if you're talking about like a protein that has its functions in the cytoplasm, then usually you're going to be talking about like folding in the cytoplasm. So there's a lot of places that proteins can fold.
Okay, thank you.
Yeah, does anyone have any other questions? Okay.
Are you able to post the PowerPoint after?
Yeah, I can share it with you guys.
Thank you.
So yeah, we're gonna go to cell structure and function. And the thing is, I didn't make any slides on the organelles specifically, just because you can easily memorize those. And so the plasma membrane, first, it's based on a fluid mosaic model, which means that you have hydrophilic heads that are outside, and you have hydrophobic tails on the inside. And so basically, you have which are made of phospholipids, and then you have like dissolves like, which means that hydrophobic protein, whatever, can basically go through plasma membranes. But if you have a polar molecule, you are not able to move through the membrane. And so you have like transport proteins that allow movement of hydrophilic or your polar molecules across the membrane. And so I think it's better to know that there are like two types of proteins. You have peripheral proteins, which basically are bound to the surface of the membrane. So like they're outside the membrane and they do not penetrate the inside of the plasma membrane. And you have integral proteins, which penetrate the hydrophobic core. So they're inside the plasma membrane. And your transmembrane proteins are basically integral proteins that go through the whole entire plasma membrane. And so transport proteins are examples of transmembrane proteins. And the thing is that a lot of proteins in the cell membrane have carbohydrate chains, and that's basically used for cell-to-cell recognition, or it's basically used as a tag for recognition. And so at cool temperatures, your membrane becomes less fluid. And so your goal is obviously to combat that, because you still want your plasma membrane to be fluid to allow the passage of molecules. So what you do is you have cholesterol, which basically helps as a membrane buffer, and it allows the membrane to be more fluid at colder temperatures, and at warmer temperatures, it prevents the plasma membrane from becoming more fluid. So it's crucial to have cholesterol in cell membranes. That if you have more fatty acids that are unsaturated, which means there's double bonds, then your cell membranes are more fluid. And so these are basically your integral proteins that they're kind of inside the plasma membrane. And then you have transmembrane proteins, which span the entire membrane, and you have peripheral proteins, which is they stay on either side of the cell membrane. And so your membrane proteins have a lot of functions. They can have enzyme activity, they could be a transporter, they can help with the extracellular matrix and the cytoskeleton, cell-to-cell adhesion, and they could be receptors. So there's a lot of different functions of membrane proteins. And then you also have transport. So there's two types of transport in the plasma membrane. You have passive transport, you have active transport. And so passive transport is basically the movement of molecules down a concentration gradient. And so the thing is, it doesn't require energy, and it's basically crucial in cell membranes to have passive transport. And then you also have these channel proteins that help with diffusion of your hydrophilic or larger substances, because polar substances like water are not able to diffuse across the cell membrane. So what it does is it has a specific channel protein, and in the case of water, it's called aquaporins. And it basically helps with diffusion of water, because the plasma membrane only allows for hydrophobic substances to cross. So what you need is a channel protein that allows for polar substances to cross. And so passive transport includes both passive diffusion, which is not using proteins, and you have facilitated diffusion, which is using nanoproteins for transport, like aquaporins. Yeah, that's basically what facilitated diffusion is. It's using proteins to allow for smaller molecules and larger molecules to through the cell. And the thing is, it facilitated diffusion and passive diffusion does not require energy. We also have active transport, which is the movement of molecules against a concentration gradient, which is low to high. So, which means that requires energy. And so, a good example of active transport is the sodium-potassium pump. And so, that's crucial in neurons, in which you want to maintain a specific membrane potential between two sides. And so what you do is it basically uses ATP to pump 2 potassium ions in and 3 sodium ions out. And so what that does is it's net negative charge inside the membrane. And you don't really need to know much about a potassium pump just because neurons aren't on the test and the nervous system aren't on the test, but it's still good to know examples of both active transport and transport. And the thing is, do you have a question? Or do you guys have any questions?
Oh, okay. I have a question. Hello?
Oh, wait, can you repeat the question?
Okay, I will ask. So, in active transport, is the only way we can move more molecules is using the energy from ATP, or is there any other source for energy?
Yeah, there's a lot of sources for energy that we'll talk about later, like in cellular respiration. That doesn't use ATP, but it uses a different mechanism. Ultimately, yeah, the source will be either. Usually, most active transport mechanisms will be using ATP.
Because I've seen that Na plus ions movement can be a source for energy. Like
yeah. So you can use You can use ion gradients for active transport, like for example, a lot of proteins use like sympore or antipore. It's basically like where certain ion diffuses across, and that allows for another specific molecule to go to active transport on the other side.
Yeah. Okay.
I see.
Yeah, so it's like I didn't put an example for like a specific for what you said, like it doesn't use ATP, but definitely in plants, if you're talking about transporting sugars, that's what they do. They don't necessarily use ATP, but they use sort of ion gradient to allow for active transport. But the ultimate thing is it requires energy.
Wait, by ion gradient, like in electron transport chain for cellular respiration, is that what you mean by?
Yeah, like that. Yeah, like basically, that is a good example of not using ATP. Or in plants, they also have a lot of ways. So yeah, it's not, but most active transport mechanisms, if you're talking about like the plasma membrane, are going to mainly be using ATP. And so, so basically proton pumps also help maintain electrochemical gradients, which is used in cellular respiration and photosynthesis. And that's kind of what I meant, I think that's kind of what you guys meant of that it doesn't require ATP. And so also carrier proteins are the only type of proteins that allow for active transport. Like you can't use channel proteins for active transport. They have to be carrier proteins to allow, because it requires a changing of shape of the protein to allow for active transport. And so you have diffusion and tonicity. This just refers to how the surrounding solution affects how cells gain and lose water. So diffusion is basically just the tendency of molecules to move from higher concentration to a lower concentration, which is the entropy, because molecules like moving more disorder, and so basically that's why molecules move from a higher concentration to a lower concentration. The thing is, it uses down its own concentration gradient, so doesn't mean that one substance is going to affect how other substances diffuse. And so an osmosis is basically the diffusion of water. And so water also goes from higher water concentration to lower water concentration. So it's basically the same, but yeah. And then you have hypertonic solution, isotonic solution, and hypotonic solution. So basically, first, the isotonic solution is basically when you have equal amounts of solute concentration on both sides, which means water will not specifically move to one specific side. Like, it's basically going to be the same. So you also have hypotonic solution, which when you have a greater solute concentration inside compared to outside, and that what that leads is to water moving inside the cell. And so cells could lyse, like specifically in animal cells. You also have hypertonic solution, which is when you have water more. And you have, a greater solute concentration outside, which means that water will move accordingly with the solutes, and that cells will shrivel. And so the important thing is that plant cells and animal cells, they have different optimal, like solute. Basically, plant cells and animal cells are ideal in an isotonic solution. So basically, the amount of water going in will be the same as the water going out. But then the thing is, since plant cells and also fungi, since they also have a cell wall, the crucial thing is that they want to be turgid, which means that they want to be in the hypotonic solution. So you should also know like the different words accordingly, like lysed will be for an animal cell in a hypotonic solution, turgid for a hypotonic solution, a plant cell in a hypotonic solution. And you also want to know like plasmolyzed, which is plant cells in hypertonic solution. Okay, so also another thing I think people were also being confused about was water potential. So water potential is just when water moves. It's just a formula to determine which side water is going to move towards. And so basically water moves from a higher water potential to a lower water potential. And so, yeah, it basically determines the direction of water movement. And so you have two variables. You have the solute potential and you have the pressure potential. And the crucial thing about solute potential is that it's always negative. And there's an equation on the AP bioequation sheet, the negative ICRT. And the thing is, you don't really need to know negative ICRT because it hasn't ever really been tested. But water potential, like the actual equation, has been tested. So you should probably know the water potential equation, but not necessarily the solute potential equation. But yeah, so solute potential is always negative because water always moves to where solutes are at. So it should lower the water potential if there's more solutes on one side. And you also have pressure potential. And so pressure can be both positive or negative. And something really important is that pressure potential in an open container is 0. And so in this, look at the picture, if you dunk a flaccid cell with a negative 0.7 water potential into a solution of distilled water, which has 0, which is basically a water potential of 0, you're going to see that water moves from a higher potential to lower potential, which means that it's going to be potential of negative 0.7. And so also, I wanted to take of larger molecules like endocytosis and exocytosis. And so endocytosis is when a vesicle goes into the cell, and exocytosis is when a vesicle goes outside of the cell. And so for endocytosis, and all of these processes require ATP. So, but then the thing is, endocytosis, there's 3 types of endocytosis that you need to know. So you have phagocytosis, which is basically cellular eating, pinocytosis, which is cellular drinking, and it's basically non-specific. It takes molecules from the extracellular fluid it randomly takes molecules from the extracellular fluid. So it's nonspecific, and you have receptor-mediated endocytosis, which is also, it's really specific in this case, in the molecules that it takes up. And yeah, also another crucial thing is contractiles. I think it's important to know how some organisms osmoregulate. So in this case, you have protists. So many protists use contractile vacuoles, which allow them to pump out water, because especially in protists that live in lakes, they're faced with a hypotonic environment. So water is always going to go inside the cell. And so what you want to do is you want to have a contractile